Hi. So 20 years ago, I went to work for a company that made heart-lung machines. And I was fresh out of college with a bachelor's in chemical engineering. And I was on a team that was designing a heart-lung machine for children and infants. And this would be used in surgeries, transplants, and stopped heart surgeries in children and infants. And I was looking at the other options available at the time as part of this company, including the recent advances in artificial hearts and in other devices to help the heart pump blood, and realized how inadequate all these were, especially for children. And it was back then, around 20 years ago, that I started to think the best thing to do as a company, kind of the apex of this medical innovation would be if we could grow a heart. If we could have a company that we just sold a heart. If these children needed a heart, instead of waiting for a donor, one could be off the shelf and implanted. And I thought about this, and there are a lot of reasons why I didn't know if this could ever be done. And in the late 90s, I started running across research, and people would send me articles because I keep talking about this. In the new field that was the term was starting to be coined, tissue engineering. And these researchers and academic institutions were talking about the idea of growing organs in the laboratory, and then these organs would be transplanted into humans. And this was exciting, and I thought, this is what I want to do. And so I left my job, I applied to grad school, quit my job, worked in a laboratory and got a PhD doing tissue engineering, learning about the materials that were needed, the cells that were needed, working in a cardiac lab and learning about cardiac development and cardiac cells. Then 5 years ago, I came here to Rice and Texas Children's Hospital and started talking with the congenital heart surgeons at Texas Children's and learning more about congenital heart disease. I came up with the idea that these strategies that I have been thinking about to grow a heart could really be used in the near term to change the way that surgeons approach congenital heart disease, which hasn't changed very much in the last 30 years. So I'm going to start with a very quick physiology lesson, and don't worry, there won't be a test or anything. But everyone recognizes that this is a heart, right? So I'll make it a little bit more complex. This human heart, like all mammalian hearts, a typical heart has 4 chambers. The top 2 are atria. The bottom 2 are ventricles. The right ventricle pumps blood to the lungs. It is separated from the left side of the heart. And the left ventricle does most of the work of the heart, pumping blood out to all the major organs, the brain, all the organs, all the muscles, and the rest of the body, and the heart itself. About 8,000 pregnancies a year, is estimated, end up with a heart that looks like this. And I won't go through this as a fetal ultrasound. I won't go through all the detail, but one thing you may be able to recognize here is that there are 3 working chambers, not 4. Only 1 ventricle, and this is like a frog heart. A frog heart has 1 ventricle. It works well for a frog. It doesn't work well for a human. So this one in particular is called hyperplasic left heart syndrome. It just means that the left ventricle never developed. And a child born with a heart defect like this will not survive without a surgical intervention very quickly after birth. Now, in the early 1980s, decades of research by very pioneering surgeons came together to develop a strategy to help children with these types of defects. What will happen if, say, a baby girl is diagnosed in utero like this with a single ventricle heart defect? A surgical team will be ready right when she is born, and that surgical team will, within the first week of life, perform a very major invasive heart surgery, opening up her chest, taking all the blood coming out of her heart, routing it to supply all of the organs, and then taking a tube of Gore-Tex and shunting some blood to the lungs to keep some blood going to the lungs. Most children come out of that and live, but it's doomed to failure in about 6 months. So about 6 months of age, this baby has to go back into the hospital, have another heart surgery, cut through all that scar tissue again from the first surgery, open up her chest again, take all of the blood that's coming down from the top part of her body and direct that to her lungs, and close her back up. And most children handle this well, and she's released from the hospital. But again, in most cases, that's also doomed to failure. And when she's 2.5 or 3 years old, her health will start to decline. She'll have to go back to the hospital for a third surgery. Again, open up the chest, cutting through that scar tissue, and taking all the blood from the bottom half of the body and now routing that to the lungs. And the blood vessels don't quite match up. Surgeons will have to take usually Gore-Tex or Dacron, so just fabric, right, and build another blood vessel so that that blood can reach the lungs. And people survive recently it's gotten as high as an average of around 90% of patients who survive, babies who survive that first week, end up surviving all 3 of these stage surgeries. And studies show, depending on the country and the study, that anywhere between 70 and 90% of these patients survive 10 to 15 years out from the study. And there are patients turning around 30 now who have had these types of repairs who are still alive. There are a lot of patients, though, in their teen years and early 20s who come back to the hospital with a failing heart after all of these operations, or they have a clot forming in this newly formed blood vessel that's, again, just fabric formed into a blood vessel, and have a stroke or other problems. And the surgeons have, at this point, really run out of options. Unfortunately, one of the only options going forward is a heart transplant. Since the lungs adapt to a low-pressure environment, one of the only options is oftentimes a heart and lung transplant, which is very rare, a very high chance of mortality, and requires the patient to be on these drugs to prevent her from rejecting this other person's heart for her entire lifetime. So 5 years ago, I came to Texas Children's, and I came in this very unique environment where we took bioengineers, materials scientists, and biologists from Rice and from Baylor College of Medicine, and we put us in a lab in Texas Children's Hospital working every day with congenital heart surgeons, pediatric cardiologists, and maternal fetal specialists, so that we could try to really translate some of these latest technologies to help children. And I was working with them. I said, I think we can do much better than this. I think we want to grow tissue in the lab, and I think that this can be used to repair these hearts and cure these children instead of just changing the blood vessels to adapt to this single ventricle heart. So I said the first thing we need to do is we need to figure out what cells to use. And I've thought of this cell question for a long time. The issue is that heart cells don't proliferate. People have been successful with tissue engineering of skin and of even trachea and bladder, and those cells you can take out of the body and grow more. And the heart doesn't do that. About 10 years ago, I was a postdoc at the University of California, San Diego, and I started talking to people who I knew in the field and reading lots of articles on human embryonic stem cells. And what they'd say is we plate these human embryonic stem cells and we just grow them in general conditions. And occasionally, part of our dishes start to beat spontaneously. And so people had isolated these, shown that they're heart cells, and this was really exciting. And I thought, this is a way that I could finally try to grow heart tissue. And I knew I had to get permission and my hands on to use some of the human embryonic stem cells. At the time, I was funded by the National Institute of Health, and this was during the George W. Bush presidency, and there are a very limited number of human embryonic stem cell lines that I, as a federally funded researcher, was legally allowed to work with. But I was able to get one of those lines, get it into our lab, and grow it, and it worked great, and I plated it in the dishes the same way other people told me. And I saw differentiation, but nothing that looked like heart tissue, and nothing happened. And I tried it again, and nothing happened. And I tried it again, and I thought, I'm going to have to be very meticulous now. And I've studied for years now on how hearts develop, and I think I can recognize how hearts are developing. So I came up with this strategy where every day, sometimes twice a day, that includes weekends, holidays, I would come into the laboratory. I would take it. I grew up for about 2 months. I grew all the cells out to have millions and millions of cells. I made them all into these tiny colonies. We call them embryoid bodies. And these little colonies, I plated in thousands of colonies in many, many Petri dishes. And I would look through all of them, and I'd look at every colony. And I would use a tiny needle, and in sterile conditions, I'd scrape off the ones that weren't doing what I wanted them to do. And I would wait for the ones that were doing what I wanted to do to get a little bit bigger, and then I'd slowly lift them off. And I would dissect them apart, and I would make more colonies, and I'd plate those in another dish. And I reduced the number of dishes. And my idea was, if I got enough of a concentration of all these cells that looked like they were becoming a heart, that maybe they would start talking to each other and become a heart. And it ended up being a good idea, but let me tell you, it was frustrating. A month went by, nothing that looked like heart tissue. 2 months went by, nothing that looked like heart tissue. There was one morning, it was a Friday morning. I'm kind of a morning person, so it was about 5:30 a.m. And I was walking in towards my lab building, and one of the other postdocs was walking out, and she came running over to me, and she said, Jeff, I looked at your cell cultures last night, and she said, I've been up all night taking movies, but you have to go look at them. And I said, what happened? She said, I don't want to tell you, but you have to go look at them. I'm going home. So I went up, ran up the stairs, took them out of the incubator. I was down to 2 plates at this time, the final 2 Petri dishes. But one of the Petri dishes on the microscope. And this is the first movie that I took that morning. It's one of the greatest times of all of my scientific research. This is what 2 months before, it was just random embryonic stem cells, and they're beating by themselves in this Petri dish. And there were a whole bunch of colonies all around this Petri dish that decided that night to just start contracting and beating by themselves. I'm not doing anything to this. This is like a little tiny heart beating in the Petri dish. It's amazing. So I got some collaborators, and they worked on how to purify and isolate the cells like this, and we published that work, and I came to Rice University 5 years ago, and I'm talking with these congenital heart surgeons, these cardiologists, and I say, I think that I can help you, and I think I can make heart tissue out of human embryonic stem cells. And we talked about the cell source, and there was a lot of nervousness about that. This was at a time when we didn't know how the government would continue funding embryonic stem cell research and if it was feasible to do and get funding for. This was at a time when there were people who were reporting that these cells were immunoprotected and wouldn't be susceptible to an immune response. But surgeons said, I don't want to put cells into an infant that I don't know if it's going to elicit an immune response, if I have to use immune suppression. And they were really nervous about that. And there are reports on forming tumors if not all the cells became heart tissue, and they were concerned about that. So I started looking through other cell sources. This exact same year that all of this occurred, there was a paper that came out, and I consider this the greatest biological discovery that anybody has made during my lifetime. It came out from Shinya Yamanaka in Japan, who made the incredible discovery that if you took an adult cell and you had viruses insert into this adult cell 4 genes that are turned on in embryonic stem cells but turned off in adult cells, some of those cells would turn into embryonic stem cells. They behave just like embryonic stem cells. It's amazing. And if you aren't familiar with this, you should look up induced pluripotent stem cells, and everyone should know about this research because it is incredible. I didn't believe it, but it's actually true. And Shinya Yamanaka won the Nobel Prize for it in October of last year. And our laboratory is working on that right now also. But the surgeons and other people I'm talking to, we still weren't quite sure how to work that into the strategy with all the timing that would be needed to fix these infant hearts. And around that time, too, I started talking with some of the maternal fetal specialists at TCH. And they said to me, you know, when we diagnose a heart defect in utero in a fetus, we take a needle and we take out the amniotic fluid. And that amniotic fluid contains cells that are the fetus's cells. And we take just a couple of those cells and we look at the chromosomes because we want to know if there's a chromosomal abnormality in this case that could be causing the heart defect. And then we just throw away the rest of the amniotic fluid and all the other cells. But this lab at Wake Forest University just published that second trimester amniotic fluid contains cells that look like human embryonic stem cells. They express a lot of the same proteins. They can make a lot of the same tissues that adult stem cells can't make. And so this really excited me. I looked into the research and we came up with this plan. And here's my plan for how I think we can get away from this surgical approach from the last 30 years and start approaching very severe congenital heart defects. About 80% of these defects at an institution like Texas Children's are diagnosed in utero in second trimester ultrasound. I think we can take amniotic fluid from there, sort out the stem cells, expand them, get a lot more of them. We can use some biomaterials to make a construct that's matched to the heart, that is like what we need to rebuild in that heart, and that surgeons can go in one single surgery and reconstruct and sew these in and rebuild up the geometry of a typical heart. And this would be the infant's own cells, is the fetus's cells. So in the newborn infant, this would be the infant's own cells, so there shouldn't be an immune rejection. And this is living tissue that should be able to grow with the infant, and I think just actually cure this condition instead of just putting it off until the next surgery is available. So our laboratory, since then, a lot of people have been interested in this. We've received funding from the National Institute of Health, the National Science Foundation, and the American Heart Association to work on various aspects of this project. And we've received amniotic fluid. We're able to isolate cells from there. Here's the cells that grow from this amniotic fluid. We've characterized them, and we've published on that they're not exactly like human embryonic stem cells, and we need to use different conditions to grow them, different conditions to make them become heart tissue. We could show that we could make them into cardiovascular tissue. Is still working. That'll be the second most exciting day of my life. We're working on this, getting these beating colonies in the dish from these cells. And then we're working on the biomaterials. And I've decided that we really need composite biomaterials for this problem. We need something that's very strong. We use a plastic that will eventually degrade and be replaced by tissues secreted by cells. This plastic gives the material really a strength and allows it to be shaped and cut and sutured and molded and make up a ventricle-like shape and be sewn into a heart. And then surrounding this is proteins that are found in the heart that contain cells that attach to it. And we've now made these materials. We've put heart cells into them. I'll go through some movies of these materials. So this is something now being grown in my lab. This is a small round disc in a petri dish that's spontaneously contracting. This is a very similar material, just the edge. We're not stimulating this, it's just spontaneously contracting. Here, we can make some different shapes out of it. Here's more the edge of a material that's beating in a petri dish. And here's another one of these materials, and these can be cut and shaped and molded and sutured, and we still have a lot more work to do to put them into humans, but I think that this is technology that can be used as a one-step cure for serious congenital heart defects. I also think it can replace technologies of patching, for example, in a defect called tetralogy of Fallot, which part of the repair is often putting a patch into the right ventricle. Works very well in short term, but leads to some long-term consequences, and we think that results are better if you put in a living patch into that area and that we can help with that. And most importantly, I think that growing this tissue in the laboratory and being able to shape it really helps to get us one step closer to this vision that I still have in my career of growing and making a heart in the laboratory. So I have to, before I go, thank all of the people who have worked on this, and these are many people in my lab who right now do all the work towards these goals that we have. And then I'd like to extend a very hearty thank you to all of you for inviting me to speak here and for attending.